Hello, good morning my friends. Today we are going to deal with some databases. You all know about a database. For example, you are studying in a degree college. Each lecturer or your principal may know you by your name, your roll number, your register number. How do you arrive all this? Because of maintaining the database. So, briefly, a database is nothing but a depository of your details in a computer for conveniently using for generation of your attendance register, your nominal roll, your marks data, and also for your distribution of scholarships. So, it is a data in a college. Like this, every organization will be having some data. For example, banks having data of different customers like savings accounts, like current accounts, etc., for easy disbursement of the cash and other transactions. Like this, every department or every organization will be having data, and in every organization this century, maintaining this data in computers using different types of softwares. For example, for the generation of your hall tickets in your college, a separate set of software will be used, it may be Excel based software, or it may be a different software. That is the utility of the software in any high-end computer system. So, like this, in a biological parlance, we are also using different databases. Earlier there was no data, and the scope of research is also very limited earlier, and it is very somewhat difficult for doing the biological data. Today in biology you have encountering different types of revolution. So, what is a difference between the revolution, because it creates the computers, they can create a revolution, the software they can create a revolution. From the development of biotechnology onwards, we are in a biotechnological revolution, and it too complex to be left to the biologist alone, because today there is an interdisciplinary set of biological research. Apart from the biologist, there is involvement of computer scientists, there is involvement of information technology personnel, there is involvement of chemists, and there is involvement of biophysicists and pure physicists, like this. All this involves, and it brings about a technological revolution that is called as a biotechnological revolution. So, 4 years back, you may come across a very important Nobel laureate to India, that is Venki Venkatragavan, or popularly called as Venki. He is actually basically he is a chemist, but he worked with the biologist and achieved a very wonderful prize to India, that is a Nobel Prize. He worked on the ribosomes. This is called as a ribozyme chemistry. So, it is a popular, now it is an interdisciplinary one. So, if you observe this slide, the colors of biotechnology, why it is called as colors of biotechnology? It can be divided into broadly into 4 areas, that is a red biotechnology, the green biotechnology, the blue biotechnology, and also the white biotechnology. What do you mean by red biotechnology? It is related to medical biotechnology, a health biotechnology, a clinical biotechnology. It involves the diagnosis and biopharmaceuticals. And the second category is considered as a grouped as a green biotechnology. It is agriculture biotechnology, the bioprospecting, and GM crops, that is genetically modified crops. And also the third category is considered as a blue biotechnology, that is marine and aquatic biotechnology, and white biotechnology, that is industrial biotechnology. So, all these investigations also involves the latest concept that is called as bioinformatics. So, for bioinformatics, all the data that is needed and all the sciences within the different branches also converge to evolve as a new science. So, basically what is a bioinformatics? The development of methods and algorithms to organize, integrate, analyze, and interpret the biological data. The study of the inherent structure and flow of biological information is the essence of this bioinformatics. What are the different goals of bioinformatics? So, you have to identify the patterns and you have to classify the different sets, maybe it is from the nucleotide or protein or any different RNA. The make predictions, you have to make predictions using the technological inputs, and you have to create models. For example, you have to create for a natural DNA, we have to also create a model and better utilize existing knowledge. So, bioinformatics thus is the application of information technology to the field of molecular biology. It entails the creation and advancement of databases, algorithms, computational and statistical techniques, and theory to solve formal and practical problems arising from the management and analysis of biological data. The primary goal of bioinformatics is to increase our understanding of biological processes. It focuses on developing and applying computationally intensive techniques like data mining and machine learning algorithms. So, this is the old biology. The old biology is very time consuming. The most challenging task for a scientist is to get a good data. It is very difficult. Now, we are entered into the new biology where data is rich, and the problem is we have to make a sense of lots of data. That is, we have to get only the sensible data from the data available. It is uploading every minute, every second. Huge amount of data is uploading to the servers which are located throughout the world. So, if you compare the old and new bioinformatics or biotechnology or sciences, it can be compared as economics. In economics of scale, you have to miniaturize the less cost due to the development of more data. The multiplex, the more data is available. You have to parallelize to save time, and automate, because there is, for example, take the automatic sequencer. It can sequence any time, any number of times you want. The minimize human intervention. Thus, you must be able to deal with large amounts of data and trust the process that generated it. The second point is the scaling of the data. What is the difference? From gene separating, that is the sequencing less than 1 KB kilobytes to genome sequencing, that is more than MB or even more than GB gigabytes. So, you have to pick up and what sort of sequencing you want. What is the logic behind this? So, the difference is the hypothesis driven research is converted to data driven research, the expertise driven approach versus the information driven approach, the reductionist approach to integrationist approach. And how to answer all this? The answer is only the bioinformatics. So, you all know that the modern science is data driven. Based on some hypothesis, data is not the end goal of the science. These are some whole genomes. Maybe you are observing these news since 2000 onwards, when the whole genome was deciphered in human beings. It is compared as a man landing on the moon. After this achievement, that is man through his Apollo voyage, and he landed on the moon, it has a big history. And after that event, this is the only activity, that is the decipheration of a human genome project. This is popularly called as a GP, is endorsed like anything throughout the world. The world leaders endorse that it will make all the change, and it can create a difference. So far, we have only limited number of whole genomes. What is genome? It is a genetic mapping of whole organism. You can observe Drosophila, it is already mapped, whole genome. C. elegans, Caenorhabditis elegans, it is already mapped. The rat, it is also whole genome mapped. The human beings, Homo sapiens sapiens, it is also mapped. There are nearly about 40,000 genes mapped so far. The mouse or rice plant, it is mapped. Mosquito, it is mapped. Yeast, it is mapped. E. coli, it is a bacteria prokaryote, it is mapped, influenza virus and Arabidopsis, all these are so far mapped, and many more organisms are going to be mapped very soon. If you observe this, if you look at this graph, it is the 1975 is the beginning of this molecular biology, and after this shift from the conventional genetics, that is conventional genetics is called as a Mendelian genetics, and from Mendelian genetics the Drosophila genetics has some change between the conventional and molecular genetics, and from 1975 there is a shift. DNA sequencing was invented. It is a very popularly known as shotgun sequencing, and after that in 1990 a Cellular Genomic Center, it is a consortium of the world top countries or world richest countries like America, England, France, Japan, Russia, Italy, that means all the European countries, US and Japan, they come and made as a consortium called as a Cellular Consortium, and at one end it is doing parallelly, and at another end the National Institute of Health, that is a purely government funded organization by America, it is funding the what is called as Human Genome Project. The beauty of the Human Genome Project is at one end a consortium, that is a world level consortium, that is Cellular Genetics. It is a private public partnership organization for the first time. For the first time they have curated genes. The result by NIH, that is US government funded agency, and the Cellular Corporation is same 100%. This is the beauty of this Human Genome Project, and this has begun in during 1990, and within 10 years by year 2000 entire human genome was deciphered. So, it is a big event in the history of biology. Now, you can observe the graph, there are a lot of data coming, and it is the problem here is it is a problem of plenty. Why it is a problem of plenty? Because you have huge data every minute, every second, huge data is coming up to the servers, and it is the scientist work to curate, to identify which is nice, which is good, which is better. There is some nuisance data is also there. So, a able biologist can identify this one. There is a lot of funding, there are a lot of publications are there, and the growth is enormous. If you observe these growths, the growth of GenBank, growth of European Molecular Bank, growth of DDBG, everything is generated, and every consortium is also funded with a huge amount of money. So, here friends, you have to choose your field, the modern biotechnology, bioinformatics, systems biology or any other emerging field, because there is no crunch for funds. You can get funds throughout the world, particularly the fundraising nations like US, Germany, Europe and Japan is also funding more for this molecular and biotechnological and bioinformatics science research. So, why I choose this topic today is to inculcate a habit of browsing the internet. It is very simple. Bioinformatics is a not big science. It is very simple. Simply you have to get internet connected. Some colleges are also having this internet connected, NMA ICT connections are there. You just simply browse. I will show you how to browse and how to get data, how to download, how to retrieve data in the coming slides. Okay. So, please be focused, particularly the science students, the future is yours, particularly the biological students, the future is yours, because so far only engineering field, information technology technologists and computer scientists are involved in this field. But for a bioinformatics, a biology student is more suitable because you know all the intricacies of what is a cell, what is a virtual cell, what are the cell organelles, what are the components, what are the mechanism that is, what is the actually the crux of the problem, that is the protein synthesis. It is actually the crux of the problem. It is called as the central dogma. That means from DNA you have to get mRNA, and from mRNA you have to get proteins. So, all these bioinformatics will move on this only in the virtual cell. So, this is used people, that is the biological science students have more future, have growth in this field. So, my sincere advice is please choose this field to grow. So, this is the graphs also shows how funding, that is NIH, National Institute of Health, that is US sponsored funding, more grants are available there. And the Medline is you can also see that how the publications are moving ahead, more publications from the bioinformatics side. So, bioinformatics is, as I said, it is evolving science. It is a new science, but evolving. It has its own roots. It can be called, it can be created as a computational biology. It can be called as a systems biology. Systems, it includes the systematics. So, now coming to the actual point, what is bioinformatics and what can it do for you? So, most research has a growing computational aspect to it. No matter what you do with every project, being tech savvy gives you an advantage. Boundaries between disciplines are blurring. The interpretation of data to make sense, drive information and knowledge, plan better experiments. You have data, you have information, and it will become a knowledge. So, now we are in the knowledge society. Different tasks of bioinformatics. What are the different tasks? The bioinformatics is, as interdisciplinary science, has to pick up, provide and apply the appropriate mathematical tools needed for tackling problems of systematic biology. They develop appropriate algorithms and implement them as effective computer programs. Provide a suitable knowledge basis to specify the application of the developed tools. Apply the developed tools onto biological problems from basic and applied research. Provide the required technical solutions for handling large amounts of biological data. What are different popular applications? There are so many applications. By as time there is the positive time, I will show you some of the applications. Phylogenetics. You all know that there is a classification popularly called as the binomial classification. When the lineage proposed, there is only about 1.5 million species identified, but many species are not catalogued. So, there is huge increase in the population. There are some new species are also identified. A recent publication of IPOL publication is a very popular referenced publication sought after Nature publications. It shows that there was 8.5 million species that are available. These are identified, but not so many species are documented even with the lineage classification, that is binomial classification. There is a growth. You have to classify at a molecular level using the bioinformatics. So, I come to that topic also in the coming slides. And genome sequencing, this is the essence of sequencing. There are different types of techniques, I will show you. The in silico molecular biology, the analysis of proteins, proteomics applications, annotations of genes and proteins, pharmacogenomics, computational drug design, microarray data mining, knowledge modeling, pathway modeling, and interactomics, and many more. So, I would like to explain one or two areas of applications, particularly basing on the databases. Here you can observe the evolutionary studies, the phylogenetics. As I said, the binomial nomenclature is outdated and old nomenclature. Still we are many biologists following that, of course, but there is a need to classify at a molecular level. So, as I said, there are 1.7 million species are available so far. So, only 1.5 million species were catalogued, that too only on the binomial nomenclature, the following the lineage. What about the molecular? What do you mean by that? Means you have to sequence each of the organism, that is whole genome sequencing. There is a huge responsibility that is laid on the shoulders of the biologist. So, come and integrate all the information scientist, the biologist, the chemist, everyone has to work on this, because it will take some more years to classify the organisms at a molecular level. And you know that this is a molecular phylogeny. This is also based on the different proteins, alpha, beta, orthologs, paralogs, homologs, etc. So, there is a lot to do, lot of research to do, particularly the biological students has to involve in this. At least a classification, if you take the classification, is a very wonderful area. This is the gene protein relationships, different organisms, different proteins from different organisms, right. As I said, the homologs, paralogs and orthologs. Coming to the database, the crux of the problem. What is a database? I said, I quoted the college experience or college database and a bank database. Like this, any organization, any association, any institution will be having a database. Here a database is a biological database. So, a database is an organized collection and storage of information in a computerized format. This biological database includes a gene sequence, protein sequence and protein structure. So, basically 3 database we are talking about, that is gene sequence, very popular. Through gene sequencing only we have the human genome project, the protein sequence and protein structure. Why these 3 are important? So, what is the aim of a database? Particularly the biological database. I am speaking about the biological database. Now, you are well versed with the database. What is the database? You know. Now, I am coming to the biological database. So, to deposit and store data, to annotate data, to check and compare redundancy in data, to retrieve data, to analyze and interpret data. These are the basic aims of a database. Different databases and the classification of database. You can observe a nucleotide database. A nucleotide database can be called as a primary database. There are primary databases, secondary databases and also composite databases. A nucleotide database includes in a server called GenBank and EMBL and DDBJ. The GenBank is maintained by US government, that is mostly predominantly America. Of course, a data can be uploaded from anywhere of the world, but it is purely maintained by US. And EMBL, it is called as European Molecular Biological Laboratory. It is a consortium of European countries and it is popularly called as European Data Bank. And DDBJ, it is a DNA data bank of Japan. So, these 3 are involved in the primary databases, that is popularly called as a nuclear database. And second one is a protein sequence database. It is having different servers like SwissProt, UniProt and PAR, that is protein information resource. And coming to the third category, that is the protein structure. Earlier you have nucleotide database and protein sequence database, it is a different, that is a structure. For this you are using different softwares like ROSMOL. So, for the visualization of the structures, PBD, MMDB, MSD like this. And a secondary database, that is having different servers like ProCite, ProDom like this, PFAM. And the classification database, the analysis tools utilities like Clustla, BLAST, FASTA, WBLAST. Okay, I will discuss about the BLAST in the coming slides. So, these are different classifications of databases. Okay, where do the data come from? There are different curators from throughout the world. The curators can upload the raw data which is actually curated in the wet labs. There is a difference between wet lab and dry lab. You, the people using the internet, using the servers, using retrieving the data, it is can be called as a dry lab. And the people who are originally working with the organisms, the cells, the manipulations, etc. in the laboratories, it is called a wet lab. So, the wet lab, a person, a scientist can upload this data. This is deposited in the primary databases, which is located in NCBI, that is what you call GenBank, US, European Molecular Biological Laboratory, and also DDBJ, DNA Data Bank of Japan. So, this information is consolidated there, and from this different scientists or different innovators can extract or can retrieve for their interpretation. It is only interpretation. So, ultimately you are having, you will be looking at a new knowledge. So, these are as I said, the primary biological databases. These left side are nucleic acid, the right side protein. The NCBI, and what is NCBI? This is a National Center for Bioinformatics. This is located in US. EMBL, as I already said, European Molecular Biological Laboratory, GenBank, and DDBJ, DNA Data Bank of Japan. So, all these servers, they primarily look at for the nucleic acid, and if you want to go for proteins, there are different servers like PIR, that is a protein information resource, the SwissProt, UniProt, Tremble, that is a trans-EMBL, PBD, MMDB, and MSD. So, these are again the nucleotide databases. For example, EMBL, align for multiple sequence, the pair based sequence, alignment databases, so different varieties. So, mutations, IMGT, immunogenetics database comprising IMGT, Elysium, like this. So, there are huge number of database. Of course, I can give you one address of a blog spot or blog. If you go for www.zoologyprgc.ac.in, .blogspot.in. I tell you once again, www.zoologyprgc.blogspot.in. If you go into this blog, you can have at least 301 links, website links for this bioinformatics, and also you will find A to Z bioinformatics glossary. This is very useful for particularly the biotechnology and microbiology students. You just search, it is readily available. Just yesterday I uploaded in this blog, zoologyprgc.blogspot.in. In this blogspot or blog, you can have different varieties of material text about the databases, about gene sequencing, and you also A to Z glossary on bioinformatics, and you are also having 301 website links for bioinformatics. You please go through this blogspot. Okay. Coming to this is a home page of NCBI. What is NCBI? It is the National Center for Biotechnology Information. This is a home page. You can read this home page, just go through this. When you have the internet facility in your college, just type in the address bar NCBI, you will have this home page. You have all resources, NCBI name, literature, DNA and RNA, proteins, sequence analysis, genes and expression, genomes, maps and markers, domains and structures, genetics and medicine, taxonomy, data and software, training and today. One important, one beautiful aspect of this is you can learn on your own. There is a tutorial, there is a training in NCBI homepage, homology, small molecules and variation. Coming to this, here at this dialog box we have all databases.